There is some regulation of glycolysis which occurs at the level of pyruvate kinase. So this is the last step in glycolysis. If we shut down pyruvate kinase, we will favor the running of gluconeogenesis. Most of these regulatory enzymes for reciprocal pathways are sensitive to two things: the energy charge within the cell, where we look at the relative balance of ATP versus ADP and AMP, and also we can consider the amount of NAD plus versus the amount of NADH. And then we have the second major factor, which is hormonal control. And hormones affect primarily the phosphorylation state of cellular enzymes. Regulation of pyruvate kinase is greatest on the liver isoform of pyruvate kinase. So pyruvate kinase activity is stimulated by fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is an early glycolytic intermediate. And now you know that that is formed by phosphofructokinase 1. Fructose 1,6-bisphosphate activates pyruvate kinase by feed forward activation. So think about the glycolytic pathway like an assembly line. Phosphofructokinase is very early in glycolysis, and pyruvate kinase is the last enzyme. Think about these as workers on an assembly line. PFK1 has been very, very, very active and has made a ton, right? This is a ton of fructose 1,6-bisphosphate. Now, it's this amount of fructose 1,6-bisphosphate is going to have to be eventually processed by pyruvate kinase once it has been through, right, all of the other glycolytic enzymes. And it's going to be in the form then of phosphoenol pyruvate, right, by the time it gets to pyruvate kinase. So it's almost like if you have an assembly line and this worker up here is yelling down the line to the workers at the end saying, hey, get ready, because here it comes. And this fructose 1,6-bisphosphate will feed forward to activate or increase the activity of pyruvate kinase. Pyruvate kinase is inhibited by ATP, which is a high energy charge indicator within the cell. Pyruvate kinase is inhibited by phosphorylation. And that means that if a phosphate is added to pyruvate kinase, then pyruvate kinase will be shut down. Pyruvate kinase is stimulated by insulin and inhibited by glucagon. So let's think about these enzymes, insulin and glucagon, which you probably, well, maybe you need a refresher on from AMP2. Glucagon is the help my glucose is all gone enzyme, right? So glucagon, help my glucose is all gone. Glucagon activates metabolic pathways which increase the levels of blood glucose. And so glucagon will act to shut down glycolysis and to upregulate gluconeogenesis. Insulin is released by the pancreas when blood glucose levels are high, and insulin will drive activation of metabolic pathways to reduce blood glucose. So insulin is going to increase activity of enzymes in glycolysis and glycogenesis, so the formation of glycogen. Take a moment, pause the video, and read these learning objectives. The enzymes of glycolysis and gluconeogenesis don't sense the hormone directly. These enzymes are being affected by phosphorylation state. Let's take a look here. I'm going to draw a line. So we really have two different panels to consider here. Let's look at the regulation of phosphofructokinase 2 versus fructose bisphosphatase 2. So PFK2 is going to act to increase glycolysis activity by increasing the amount of fructose 2,6-bisphosphate in the cell, which increases the activity of PFK1. Fructose 2,6-bisphosphatase is going to deplete the amount of fructose 2,6-bisphosphate and therefore reduce the activity of PFK1 and reduce the activity of glycolysis. Don't skip steps, okay? If we look at the bottom panel here now, the key thing to remember is that insulin drives glycolysis because insulin is trying to act to reduce glucose levels. So we're going to cause glucose to get burned up in glycolysis. Glucagon, the help my glucose is all gone hormone, is going to drive gluconeogenesis. So we're going to make more glucose because we need more glucose. Insulin acts to drive overall, and I'm putting overall, dephosphorylation in the cell. Glucagon acts to drive overall phosphorylation in the cell. Phosphofructokinase 2 and fructose bisphosphatase 2 are opposite sides of the same enzyme. So these together form a bifunctional enzyme. One portion of the enzyme acts as PFK2, and the other portion of the enzyme acts as a phosphatase. Insulin drives a phosphorylation cascade, which leads to activation of an enzyme known as protein phosphatase 1 or phosphoprotein phosphatase 1, PP1. When phosphoprotein phosphatase is active, we will have dephosphorylation and activation, see the OH group here? Dephosphorylation of this complex and activation of PFK2. And so PFK2 is going to phosphorylate fructose 6-phosphate and form fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate then activates PFK1, right? So let me find a way to draw this in. So I'll erase this. PFK2 is going right from fructose 6-phosphate. We have the input of ATP, and what do we get out? Fructose 2,6-bisphosphate plus ADP. Okay, that's what's happening here. And then this goes on to activate PFK1 and glycolysis. When glucagon is active on the cell, there is a phosphorylation cascade, which leads to activation of CAMP-dependent protein kinase. I'm going to put PKA, protein kinase A. Protein kinase A adds a phosphate to this enzyme complex. Here's the phosphate. When this bifunctional enzyme is phosphorylated, now fructose bisphosphatase 2 is active. When fructose bisphosphatase 2 is active, we're reducing the concentration of fructose 2,6-bisphosphate because it's becoming dephosphorylated, right? And this phosphate leaves as PI. And so by dragging down the concentration of fructose 2,6-bisphosphate, we reduce the activity of PFK1, and therefore we reduce the activity of glycolysis. I tell students to memorize one thing. So we're looking at reciprocal control here of insulin versus glucagon. Make a map Make a map in your mind about what is happening when insulin is active on the cell, and then know glucagon, it's essentially opposite. Another way to look at this, and this is an image taken from a literature report, when blood glucose is low, glucagon rules, right? So help my glucose is all gone. Glucagon is a peptide hormone. And so it is a water-soluble hormone. It binds to a receptor in the cell membrane, and that is a G protein coupled receptor. When we have binding of glucagon, we then release the G protein from the hormone receptor. The G protein diffuses laterally along the interior leaflet of the cell membrane and is going to activate adenylate cyclase. Adenylate cyclase is going to cyclize ATP to form cyclic AMP. Then cyclic AMP is going to bind to and activate protein kinase A, right? Protein kinase A is a cyclic AMP dependent kinase. And PKA is going to phosphorylate phosphofructokinase 2, fructose bisphosphatase 2 bifunctional enzyme, and will cause the fructose bisphosphatase activity to be active. And so we're going to have a reduction in the level of fructose 2,6 bisphosphate. And so you can see here that we get less fructose 2,6 bisphosphate. So there's no PFK1 stimulation and glycolysis is reduced. When insulin is active on the cell, the opposite will occur. When insulin, which is, I don't know why it's not shown on the image, insulin leads to activation of protein phosphatase 1. Protein phosphatase 1 dephosphorylates this complex. I'm going to put an OH here so it matches the image that we just looked at. PFK2 is now active, and we're going to lead to increased levels of fructose 2,6 bisphosphate, which stimulates and activates PFK1, and so glycolysis is increased. Gluconeogenesis in mammals cannot use the carbons from fatty acids. So we cannot use the carbons right in the fatty acid tail from fatty acids, but we can use the glycerol backbone. So we can use the carbons from glycerol in order to build glucose. Let's look at this. In glycolysis, we can form pyruvate. And then there's a large enzyme complex called pyruvate dehydrogenase, which we abbreviate PDH. Pyruvate dehydrogenase is going to decarboxylate pyruvate. The remaining two carbons of pyruvate will be coupled to coenzyme A to make acetyl-CoA. In the turning of the TCA cycle, these carbons are going to be thrown out as CO2. And so the carbons of pyruvate are going to be thrown out as CO2, as CO2, as CO2 before we have formation of oxaloacetate that we then use in gluconeogenesis. When we oxidize fatty acids through the process of beta oxidation, again, those carbons from the tail, right, of the fatty acids enter into the TCA cycle as acetyl-CoA, and we throw them out as CO2. So they never make their way into oxaloacetate over here. Likewise, where we have lactate and other amino acids, I'll put like alanine, being converted by LDH or transaminases into pyruvate, these carbons of pyruvate are going to not make it over to oxaloacetate and then into glucose. However, if we transform pyruvate to oxaloacetate by pyruvate carboxylase, now we can capture those carbons into glucose. As far as taking acetyl-CoA, which could be formed from fatty acids, and saying, well, how come we can't convert acetyl-CoA back to pyruvate? It can't be done. Pyruvate dehydrogenase complex enzyme is irreversible in humans. Note that other amino acids can flow into the TCA cycle after the decarboxylation steps. So there's 2 decarboxylation steps in TCA cycle. Amino acid skeletons can flow in beyond those points and make their way into the carbon skeleton of oxaloacetate, which we can then use to form glucose. These amino acids are known as glucogenic amino acids. The approach to gluconeogenesis differs in different forms of life. We don't really care so much about the plants. Let's take a look here in animals, and then we have this common area between animals and plants. So this is where we are as human beings. We're this panel and this panel. So we cannot use the acyl chains or the fatty acyl chains of triacyl glycerols to make glucose, but we can use the carbons of the glycerol backbone of triacyl glycerols. We can use the carbon skeletons of glucogenic amino acids. And who are glucogenic amino acids? All amino acids, but not lysine or leucine, are glucogenic. And if you want to know what TCA cycle intermediates these different amino acids can be converted into, right, and then work their ways over to oxaloacetate, the lists are here for you. You do not need to memorize this, but you should memorize this. All amino acids, but not lysine and leucine, can be used to make glucose, right? And so here you see the conversion of lactate to pyruvate. This is done by lactate dehydrogenase, and then pyruvate is able to be converted to oxaloacetate by way of pyruvate carboxylase. And then, of course, right, we can take oxaloacetate and convert it to phosphoenol pyruvate by PEPCK. And then, right, we're going to work our way up through gluconeogenesis to glucose 6-phosphate, and glucose 6-phosphate can enter into a lot of different pathways within the cell. Pyruvate can be used to replenish oxaloacetate, and that is done via the pyruvate carboxylase reaction. Pyruvate carboxylase is present inside the mitochondrial matrix. This enzyme we just looked at in gluconeogenesis. We'll get some more particulars of it now. So here is bicarbonate, right? HCO3 minus. Here's bicarbonate. Where does this come from? It's an integral component of the bicarbonate buffer system. So there's no real shortage of bicarbonate within cells. ATP is needed by pyruvate carboxylase in order to activate bicarbonate. So this is the portion of the molecule that was bicarbonate. Elevate the energy level of bicarbonate so that the carbons of bicarbonate can be covalently coupled to biotin cofactor, right? Also known as vitamin H. So there's our biotin cofactor, and this is on a lysine amino acid, and lysine can flex. So this biotin cofactor is kind of like an arm that can flex from one side to another, and we can pass this bicarbonate from one active site on pyruvate carboxylase to another. So we have one site for the phosphorylation of bicarbonate to form carboxyphosphate, and then we have a second active site where we are going to have transfer of that bicarbonate to pyruvate to form oxaloacetate. So here's the one carbon from your bicarbonate. So biotin is a one carbon carrier. Here's the image from your book, and it's nice because it shows more detail of the enzyme. So here's catalytic site 1, where we have the coupling of phosphate to bicarbonate. And this is going to form carboxyphosphate, which is a high energy compound. It is a mixed anhydride, so it's a high level compound. It's a mixed anhydride. The second part of the reaction is occurring at catalytic site 2, right, which is not really shown here. Catalytic site 2 is where we have the transfer of to pyruvate to form oxaloacetate. This is a nice diagram too because you can see the linkage here of the biotin cofactor to lysine. And lysine has this chain of CH2 units. Normally we would have a amino group here, so alpha, beta, gamma, delta, epsilon, amino. There's an epsilon amino side chain. These CH2 groups here are very flexible because there's free rotation about all of these bonds, and so we have this biotin 1 carbon carrier on a flexible arm, and we can passage 1 carbon units from this active site over to this. This image is showing you the passage of CO2 to pyruvate. Right, so biotin is decarboxylated. Pyruvate is converted to its enolate form. Why? The enolate form has a higher energy state. And here you see the combination of the CO2 to the enolate form of pyruvate. This coupling is non-enzymatic. Do you see that? There's nothing here on the enzyme that is affecting this. This is non-enzymatic. What does the enzyme do? The enzyme helps to elevate the energy state of pyruvate to the enolate form, and then we have this non-enzymatic coupling of CO2 to pyruvate.